far have we come? My name is Padraig Dixon, and I'm a research fellow with the Centre for Personalised Medicine here in Oxford. The centre is a partnership between Oxford Centre for Human Genetics and St Anne's College, and it is a communication, engagement and research vehicle for students, academics, clinicians, the public, and policymakers to explore the benefits and challenges of personalised medicine. And the centre's principal aim is to explore personalised medicine from a range of perspectives. We are therefore really delighted to welcome Dr. Emma Anderson from UCL. Emma is Associate Professor of Epidemiology in the Division of Psychiatry at UCL, and her work focuses on the application of novel causal inference methodologies to identify modifiable causal risk factors for Alzheimer's disease. Emma is a UKRI Future Leaders Fellowship holder, and her work on the fellowship will extend her previous work in the space by identifying genetic and environmental determinants of vascular dementia. So we're really delighted to have Emma give this seminar. If you have a question, please put it in the chat, and at the end of Emma's presentation, we'll have a chance to cover as many questions as possible. So without further ado, I will ask Emma to share a screen and kick off whenever you're ready. Thank you, Emma.
No worries. Can you see my screen?
Yeah, that's perfect. You're a little bit quiet. If there's anything you can do about that, we can see you very well and hear you pretty well.
Just speak louder. Is this okay?
That's perfect. Thank you. You're good to go.
I don't think I've ever had anyone in the history of my life tell me that I'm being quiet. So yeah, that's a first. Thank you so much. And thank you for inviting me to talk to everybody today. So yeah, I wanted to talk about kind of what we understand about the causes of dementia. And I wanted to clarify at the start, when I say causes, I am talking about essentially risk factors. So things that we can modify to alter our risk of dementia. And I guess the talk is intended to be a bit of a broad overview. I'm not going to go into too many specifics about methods or too much detail about any particular study. I just wanted to highlight kind of the key challenges that we face in dementia prevention research, and just highlight kind of why we've had such a tough time, I guess, figuring out what causes it. So I wanted to start by thinking about why we should care about dementia and how big of a problem really is it. So the kind of best estimate at the moment is that there are around 55 million people living with it globally. We've had massive rises in developing countries as people start to live longer, and not many people know this because I think most people think it's either CVD or cancer, but dementia has actually been the leading cause of death in the UK, apart from COVID, since 2015, which I thought was really surprising. It's really expensive, so it costs the UK alone about £26.3 billion a year. And just for some context, that's the annual energy bill of every single person in the country. So it's quite expensive. And 2/3 of the cost of dementia is paid by the people that have dementia and their families, either in unpaid care or in paying for private social care, which is a bit of a travesty. So the term dementia itself is actually just an umbrella term that refers to the range of cognitive impairment symptoms that we typically think about. So things like memory loss, loss of executive functioning, so issues with things like planning and problem solving. And these symptoms can have many different underlying causes. Some of them are shown here. It's kind of commonly thought that Alzheimer's disease is the most prevalent form of dementia, followed by vascular dementia. But we actually know from post-mortem studies that most dementias are, in fact, what we call mixed dementia. So patients will most often have more than one type of pathology in their brain at any one time. And figuring out which is the kind of key driver of that cognitive impairment can be quite tricky. So that leads me on to thinking about how dementia is diagnosed. The gold standard diagnosis is a neuropathology exam at post-mortem. So that's, you know, the most accurate way, but is obviously not particularly helpful for understanding disease in living people. In living patients, in an ideal world, we would use probably MRI scans and potentially a sample of cerebrospinal fluid to look at biomarkers of things like beta amyloid and tau. But those are both expensive and also quite invasive. So in reality, it's a bit of a postcode lottery which examinations you get depending on what resources are available in your local memory clinic. So usually it's really standard to perform a series of cognitive tests, and most people will get some sort of scan, usually a CT scan, just to rule out any other major causes of impairment, so things like tumours and big strokes. So before I go on to what the evidence says about the causes of dementia, I thought it would be useful just to be kind of upfront at the start about what the key challenges are in dementia research, because I think knowing that is kind of key for appraising studies that that are published. So I'm assuming that we all know that correlation doesn't necessarily equal causation, and this, as we know, is particularly problematic in observational research. So confounding is always a huge issue. I'm sure I'm probably preaching to the choir. Most people here will know what confounding is. It's also not unique to dementia research, but it is, one of the challenges that we face. So we know that traits or phenotypes tend to cluster. For example, we know that people who have better diets tend to also be more physically active, less likely to be obese. So the two kind of key issues are, firstly, knowing all of the confounders. Do we ever really know kind of all of them, and can we measure them all? And secondly, even if we could hypothetically measure all confounders and adjust for them in our analyses, we still have issues with residual confounding because of measurement error. And across all confounders, that could add up to explain away, not insignificant magnitudes of effect that we might observe between a risk factor and an outcome. So although it's, not unique to dementia research, it is a problem that we face. The second big challenge, again, a really big one generally in observational research, is reverse causation. So reverse causation is really problematic in dementia research in particular, because you will probably know that dementia has a prodromal phase that can last over 2 decades. And so this prodromal phase is kind of the phase of the disease process where the pathology has started accumulating, the disease process has begun, but the individual is not aware of that, and they are not yet experiencing any kind of obvious or clinically significant cognitive problems. We have relatively recent evidence to suggest that this disease process may even begin in childhood, so it could potentially be lifelong. And the issue with reverse causation is that it can make us think that a risk factor is causing the disease, when actually the disease or the prodromal dementia, these kind of early pathological changes in the brain, are causing the risk factor. So a good example of this is for depression. I think the jury is still out on whether we think that depression is likely to be a causal risk factor for the onset of dementia. I think the current thinking is more sort of down the avenue that it is likely to be reverse causation, and that those early pathological changes are likely to make somebody more at risk of depression. And it's a similar story for social isolation. So is social isolation going to make somebody more at risk of being diagnosed with dementia, or are those early brain changes making somebody more at risk of being socially isolated? So the third challenge that I wanted to think about was randomized control trials. You might be thinking, well, if we have issues with confounding reverse causation, why don't we just do some RCTs to figure out, what the causal risk factors are? But because of that long prodromal phase, it's really tricky to randomize somebody to, say, a physical activity intervention pre, those pathological changes beginning, and then follow them up for, say, 20 to 30 years, maybe even longer, to see whether they do or do not end up with a dementia diagnosis. So it's just unfeasible really to run RCTs for dementia prevention for, many risk factors. There are also a whole bunch of risk factors that we just simply cannot randomize. So we are interested in things like air pollution. Some things just, as you know, don't lend themselves well to RCTs. So it's not the kind of, great answer that we were hoping for. The fourth challenge that I wanted to highlight is survival bias. And again, this is problematic for lots of diseases, but it's really problematic for dementia research because it's a really, really late life disease. So even for things like cardiovascular disease, the magnitude of survival bias is likely to be much lower than it is for a disease like dementia, where the average age at diagnosis is around 80. So survival bias, as I'm sure you're aware, is a form of collider bias where essentially people are dying prematurely of the risk factor that you're interested in studying. So you get these spurious associations between the risk factor and the outcome. A really good example of that is smoking. So we can often observe that smoking looks to be protective for dementia, but actually what we think is happening is just that people that smoke tend to die essentially before they get a dementia diagnosis. So it looks like not smoking is leading people to go on to get dementia, but actually it's just that the smokers kind of die before they are able to get the dementia diagnosis. And yeah, survival bias is an issue that affects kind of most study designs. There isn't really a study design that is immune to survival bias. It's hard to get around it in that design phase of the study. And really, we just end up applying statistical methods to kind of undo that bias during the analytical phase. And the final challenge that I wanted to highlight is this kind of heterogeneity in the outcome. So I talked about how most cases of dementia are mixed and how we can't really diagnose dementia very accurately while people are still alive. And this can cause problems, again, in most study designs because people get misclassified. And this is problematic if the risk factors differ for each kind of subtype or cause of dementia, because the effects that we observe in any given study will just depend upon the proportion of dementia types that we have within our sample. So that can lead us to seeing quite conflicting results across studies for the same risk factor, and it all just kind of gets a little bit messy. So now we are more aware of the kind of challenges, I wanted to move on to thinking about what causes dementia. And the main focus of the talk, as I said, is going to be around these modifiable risk factors, but it would be remiss of me not to mention the ApoE gene, because it is by far and away the strongest risk factor that we know of for dementia. So ApoE is a gene that sits on chromosome 19. There are 3 forms of this gene that you can see here on the right-hand side, and they each essentially just reflect the underlying structure of the gene, so the way that the proteins are structured. ApoE4, this one highlighted in red, is the form that confers the highest risk of Alzheimer's disease. So in fact, it's about a 12-fold greater risk if you have 2 copies of ApoE4 compared to if you had 2 copies of ApoE3, which is the one in the middle. So ApoE3 is considered kind of neutral, if you like, and is by far the most common form of the gene. So it's about 95% prevalence worldwide for having at least 1 copy of ApoE3. This kind of bad form of the gene, ApoE4, the prevalence is about 28% for having 1 copy. And then lastly, we have E2 up at the top here, which is kind of the protective form of the gene, if you like. So it confers between 40 and 80% protection when you compare it to ApoE3. And this one, annoyingly, is the most rare form. So that's about a 14% prevalence of having at least 1 copy. And I guess it's worth mentioning that lots of the associations that we will talk about have been shown in observational studies to interact with ApoE. So there is some evidence that ApoE status can change the effect of, for example, smoking or alcohol consumption or physical activity on dementia risk. So it's almost like an effect modifier. So for modifiable risk factors, a good starting place where people kind of tend to gravitate to is the Lancet Commission. So the Lancet Commission is essentially a huge review of the evidence for dementia risk factors, and they came up with 12 risk factors that they kind of estimated if we intervened upon those, we could prevent around 40% of all dementia cases. So they sort of go through the life course. So we've got educational attainment in early life. Then you've got these kind of midlife risk factors of hearing loss, traumatic brain injury, hypertension, alcohol consumption, and obesity. And then what they deem as being kind of later life risk factors. Although I've always questioned why some of them aren't in the midlife group as well. But yeah, for the later life ones, we've got smoking, depression, social isolation, physical inactivity, air pollution, and diabetes. And yeah, like I say, they kind of estimated this population attributable fraction, which suggested that if we could intervene on all of them, we could prevent about 40% of all dementia cases. But the key thing that I wanted to note here is that there is a really quite strong assumption underlying all of these risk factors that they are causal. And this assumption is mentioned in kind of a really small sentence, which is buried away in the limitation section of the review. But if something isn't causal, then intervening on that risk factor is unlikely to have any meaningful effect on dementia. And given the challenges that we've just spoken about in assessing causality for these risk factors, I think our confidence that these are all causal risk factors is relatively low. So the WHO has a slightly different opinion on which of those risk factors are likely to be causal and therefore good targets for intervention. So they had a panel review all of the evidence for the key hypothesized risk factors, and then they made recommendations for dementia prevention based on the results of that review. So based on the strength of kind of all reviewed evidence, I guess it's worth saying that they didn't look at traumatic brain injury and air pollution in their review. But they deemed that for hearing loss, depression, and social isolation, that the evidence was kind of too ropey, I guess, to make any sort of recommendation around dementia prevention. They then made what they called conditional recommendations for hypertension, alcohol, obesity, and diabetes. And the conditional recommendations are essentially where the evidence isn't great, but they explicitly say that because the evidence is good for other diseases, so has other known benefits, so for things like CVD, we should still intervene. But it doesn't really tell us much about dementia etiology. They are very clear that, you know, it's more of like, we think this is important, but the evidence is quite ropey. And then they make kind of strong recommendations for educational attainment, smoking, and physical inactivity. So yeah, I guess it's kind of interesting that these two bodies that have reviewed the evidence independently have come up with quite different recommendations about dementia prevention. So as I said earlier, RCTs for dementia prevention are really tricky, and most RCTs today actually have failed to identify an effect. I think it's like over 98% of dementia RCTs have not identified any effect. There are lots of potential reasons why, but one reason that we think is intervening too late. So we are thinking that we need to kind of go before this prodromal phase. So that means ideally intervening in, you know, people's 40s and 50s, and then following them up to a diagnosis, which, as we said, is not always feasible. It also depends what you're measuring. So, you know, whether you're looking at all-cause dementia, whether you're looking at Alzheimer's disease specifically or vascular dementia specifically. And it also depends on the intervention. So we have these kind of big multi-domain interventions where they're, you know, doing like a multi-pronged approach at lots of different factors versus intervening on one single kind of phenotype. So this diagram here is from the FINGERS trial. And FINGERS is essentially a big multi-domain 2-year randomized control trial that included people over the age of 60 who were at increased risk of dementia. And when I say at increased risk, they used one of the risk prediction scores. I think for FINGERS, they used the CADE score. So people were randomized to a lifestyle intervention, which was around sort of nutrition and exercise. And then they did some cognitive training, as well as intensive monitoring and management of metabolic and vascular risk factors. And at the end of the 2 years, there was some evidence of small beneficial effects on cognitive function in the intervention arm, which was, you know, kind of exciting because everything else up to this point has been pretty null. But I guess it's worth saying that there are some concerns around this trial and others like it. So one of the worries that we have is about practice effects from the repeat cognitive testing. And you might say, well, you would only get practice effects in, the intervention arm. So that wouldn't explain, the difference between the intervention and the control arm. But if the cognitive training that they do is quite similar to how they test cognitive function at the end to compare outcomes, then that can lead you to see a kind of difference that is due to a practice effect and not due to there actually being an improvement in cognitive function. And there are also concerns about these kind of really small, statistically significant, but not necessarily clinically significant effects on cognition. Worries that they're not going to be translated into any actual meaningful effect on dementia risk. I guess the other thing worth highlighting is the concern about selection bias within these trials. So I think for this fingers trial, they worked out that people in the intervention arm had to do around 360 hours of engagement, and there was a big question mark around who are these people that are kind of signing up and engaging in 360 hours of intervention. Are they representative? Are we going to see that level of adherence in the real world, and are we going to see the same kind of effects on cognition being observed in the real world? And I guess, again, worth pointing out with this fingers trial that they just did cognitive testing at the end of it. The follow-up is ongoing. They have another, I think, 7 years of follow-up plan. So they will look at kind of hard endpoints for dementia diagnoses, but currently we're just looking at cognitive testing. So we can't say if these interventions are important for things like Alzheimer's disease versus vascular. We are just looking at cognitive function sort of more broadly. The other trial that I wanted to highlight is the SPRINT MIND trial. So this is another big trial, but it's of just one risk factor. So this one is of blood pressure. They had intensive blood pressure treatment versus standard care in 50-plus-year-olds with hypertension, so quite young for a trial of this type. And the trial was actually designed with CVD endpoints as the primary outcome. So it was stopped early for showing benefit. So there isn't actually any statistical evidence for an effect on dementia risk, which is one of the kind of secondary outcomes. But it potentially wasn't powered for that endpoint or didn't sort of go long enough. But it does, I think, look as if there is some evidence of a difference here between the intensive treatment and standard treatment. I guess, just if they carried on longer or if they were powered to detect an effect on that outcome, it looks as if they could have potentially observed an effect with blood pressure. But again, it's worth noting that this trial specifically was with any dementia and not specifically with Alzheimer's disease or vascular dementia. So because RCT for prevention is so tricky, I wanted to talk about a method which almost tries to mimic an RCT. And this is where most of my work has focused over the last kind of 7 years or so. So we said previously that when we measure somebody's risk factor, we do that in a kind of observational way. So we would ask if they smoke, and we say, if you do smoke, how many do you smoke? And then we said, if we look at these things observationally, we have issues with confounding. So things like educational attainment can determine whether somebody is likely to end up smoking and whether or not they're likely to end up getting dementia. And so that confounding can cause us to get the wrong answer, essentially. And we also have the issue of reverse causation bias for some risk factors, where that prodromal dementia can sort of change the risk factor and behaviors. So Mendelian randomization is essentially an instrumental variable method where you're using genetic variants almost as proxies for the risk factor that you're interested in, instead of measuring somebody's risk factor. So instead of measuring their smoking habits, we would instead measure their genetic variants for the risk factor using all available known variants from a genome-wide association study of smoking, either smoking initiation or smoking heaviness. So for this example, we have genes that are associated with whether somebody is likely to smoke at some point in their life. So those are kind of like addiction genes. And then secondly, genes that determine how heavy we smoke if we do take up smoking. And so a good example of this is the Chrona5 gene. And the benefit of using genes is that they are randomly allocated. So nothing can determine whether or not you get that Chrona5 gene from your parents except for chance. So it essentially means that in theory, it is not associated with anything other than your likelihood of smoking or how heavy you smoke. So this annoying arrow that we had before between the confounders and our exposure, that arrow is kind of broken. We've broken the link between the confounder and the risk factor because we are now using genes as an instrument. The other kind of cool thing about MR is because what gene you end up with is randomized at conception, nothing after that point, so no diseases, no kind of lifestyle changes, no prodromal dementia, can go back in time and change the gene that you were allocated. So we don't in MR typically worry about reverse causation. So essentially the randomness has kind of helped to fix two of our key problems in dementia research. So MR has been previously deemed to be nature's RCT. This has been unpicked slightly, but it's essentially just a way of comparing MR to a randomized control trial. So in a randomized control trial, we get random allocation of treatment. For MR, we get the random segregation of alleles, which leads to an exposed group and a control group, just as you would get in an RCT. Because of that randomization process, our confounders should be equal between our exposed and our control groups. And then we can go on and compare the outcomes that we are interested in between those two groups. And so I guess the key thing here is to say that, you know, we might not be able to do a randomized control trial in dementia research, like we said, because we have to follow people up for 20 to 30 years. But in MR, we don't have to do that. It's kind of analogous to an RCT, except that our genes are already there, kind of waiting to be measured. We don't have to randomize people and then follow them up for 20 to 30 years. So I wanted to give a couple of examples of where MR has really gotten it right. You might remember back during COVID, it made the news that an arthritis drug could help reduce COVID severity and death. So they ran clinical trials because it was essentially thought that this extreme immune reaction to COVID was essentially making patients more sick. And so this arthritis drug, which is an IL-6 inhibitor, essentially inhibits that extreme immune reaction. And the thinking was that then you would potentially have better outcomes for COVID patients. So they conducted RCTs, randomized people to the arthritis drug, and indeed the patients did have better outcomes. So they were less likely to be hospitalized and less likely to die. And interestingly, before that trial was even run, there was an MR study that was conducted, a drug target MR study, where you essentially take genetic variants that mimic the effect of certain medications and see how that relates to risk of an outcome. And the drug target MR that they did for this arthritis drug essentially showed exactly the same as what the trial had shown. So again, yeah, fewer hospitalizations, less severe COVID. I think they also looked at death. And this MR was actually published the year before the randomized control trial was run. So there are, I guess, examples where, provided the assumptions of MR are fulfilled, it has the potential to kind of mimic an RCT. We have another example of this, which was for the effect of selenium on prostate cancer. So for years and years, observational evidence suggested that selenium was likely to be protective for prostate cancer. And then randomized control trials were run, and they showed that if anything, it was harmful. MR studies, you know, much later when they came out, essentially confirmed that it was null. And we have a similar story also for CRP and coronary heart disease. So again, for years, we thought that CRP was causal for coronary heart disease. But the MR, along with the RCT evidence, is showing that intervening on CRP is very unlikely to affect CHD risk. So I wanted to give you an idea about how MR has fared in examining dementia risk factors specifically. So this table is based on those WHO risk factors that we spoke about earlier. And based on the kind of Lancet and WHO recommendations, we would hope that all of these MR studies, so each number here represents a study, and then we've got the references at the bottom, we would think that most of them should be in the harmful column because they have all been deemed to be risk factors for dementia. But as you can see, most of them are null, and actually some of them we're even seeing some protective effects. So for things like smoking, physical activity, there is some evidence that they're kind of going in the opposite direction to what we might expect. And the only one that is kind of consistently showing a harmful effect, and the only one that I think looks remotely convincing, is educational attainment. So the MR evidence isn't particularly great for suggesting that these are kind of indeed causal risk factors for Alzheimer's disease. And I guess the one thing that I wanted to point out here is that we are looking at Alzheimer's disease specifically and not other causes of dementia, because the Mendelian randomization studies done for dementia, they will generally use an Alzheimer's disease GWAS, where they have screened out for things like cardiovascular risk. So there is likely to be a sample of kind of mixed dementia in there, but they will definitely be enriched for Alzheimer's disease. So this table could potentially look different for other causes of dementia. We might perhaps expect that hypertension and other CVD risk factors might associate with increased risk of vascular dementia, but potentially not Alzheimer's disease. So educational attainment, I think, is perhaps the risk factor with the most convincing evidence so far. We see the same direction of effect observed in almost all observational studies and MR studies. So we did an MR study going back a few years ago now, where we essentially took genetic variants that associate with the amount of time that people stay in school, and then genetic variants that associate with levels of intelligence or kind of general cognitive function. And the idea was to see whether they independently, years of schooling and intelligence, have an effect on dementia risk. So that's this red line here on the forest plot. So you can see that more years of schooling and higher levels of intelligence associate with lower odds of dementia, or Alzheimer's disease specifically, sorry. And then the second aim was to do what we call a multivariable MR, where you essentially adjust one for the other to see if educational attainment had an independent effect on dementia risk once you take account of kind of prior levels of intelligence. And you can see here that the independent effect of years of schooling attenuates towards the null, and we lose quite a lot of power. But it looks like there is an independent effect of intelligence on dementia risk, so independent of years of schooling. So what we think is happening is essentially that years of schooling can affect dementia risk. More years in school can lower dementia risk by increasing intelligence, and it's intelligence that has that kind of direct effect, whereas the years of schooling has an indirect effect sort of via intelligence. And the cool thing about this study and other kind of MRs of educational attainment is that the results of those studies align remarkably well with other instrumental variable studies that don't use genetic variants, but they use other instrumental variables. So one of them is a policy reform, raising of the school leaving age from 15 to 16. That's been done both in the UK and in the States. And it has been shown that raising of the school leaving age improves cognitive function and reduces risk of dementia. So we've got kind of triangulation across two quite similar methods, but very, very different instruments giving really similar answers, which is kind of reassuring. We've also used MR to examine the effect of different sleep traits on dementia risk. So here we took genetic variants that have been associated with both self-reported and accelerometer measured sleep traits. And frustratingly, we found very little evidence and effect of any of the sleep traits on Alzheimer's disease risk. So these are all the sleep traits that we examined here on the left-hand side. And you can see that most of them, there's just a lot of variation around the null. But there was some evidence for daytime napping, tentative evidence, but that has since been replicated in other independent samples. So I guess, yeah, the jury's out on whether we should all just be taking more naps to prevent dementia. And then the other one that I wanted to mention was a phenome-wide association study, which was done by our PhD student in Bristol, Roxana. And this was done in the UK Biobank. So a phenome-wide association study, or a PheWAS, is essentially almost like a reverse MR study. So you take a polygenic risk score for a trait that you are interested in. Here it was Alzheimer's disease. And you essentially do a hypothesis-free scan for associations between that polygenic risk score and the entire phenome. So here we had over 15,000 variables in the UK Biobank. And it's important to note that for this study, we stratified everything by age tertiles. So we were looking at these associations in sort of young, middle-aged, and old participants. So essentially the take-home, what she run the PheWAS, and then any sort of hits that you get out of that, so associations between the PRS and the trait, then gets followed up in a formal 2-sample MR setting. So rather than it being PRS for Alzheimer's disease on the trait, you're then taking genetic variants for the trait and saying, does that causally relate to Alzheimer's disease? And the PheWAS suggested that in that kind of first initial step, that increased genetic liability for Alzheimer's disease, so a higher PRS, affected a whole kind of range of phenotypes. Things like medical history, there was brain-related phenotypes, so things like hippocampal volume, brain volume, physical activity, some blood-based measures. And I guess the key take-home message from Roxana's study was that most of those associations, firstly, tended to be driven by the ApoE gene. So when we removed ApoE from the polygenic risk score, most of these associations attenuated to the null. And the second take-home from her study was that when she followed up those hits, those PheWAS hits with a formal MR, a very, very small minority of the variables showed evidence of being causally related to Alzheimer's disease. There was only a couple. I think there was things like forced vital capacity. There was like body fat-free mass. There was about 5 or 6, I think, out of 15,000 variables that showed evidence of being causally related to Alzheimer's disease. So it looks like most of the ones that we are seeing in that initial PheWAS phase are due to either reverse causation, so prodromal AD causing those traits, or some kind of bias, so something like selection bias or survival bias, depending on how it differs across the age tertiles. And the other kind of take home is that we were observing effects of AD liability at a really quite young age. So we were seeing things cropping up in that youngest tertile, and they were aged 39 to 43 years, I think. So potentially we're seeing manifestations of the AD prodrome, you know, quite early on, which is quite interesting. I wanted to highlight a drug target MR study that has been done in dementia rather than COVID, because that COVID example was such a kind of solid example. And so I think this is quite a promising area for dementia research generally. So this is a study that was led by colleagues at UCL, and they instrumented statin use with genes that encode the protein targets of several lipid-lowering drugs. So they used HMGCR and PCSK9. And essentially, they found very little evidence that statins would affect dementia risk. And if anything, there was actually some evidence that exposure to PCSK9 inhibitors might actually increase dementia risk, which is kind of disheartening. I think I was a little bit sad that this was null, but actually it's really important, I guess, that we run these drug target MR studies and understand where there is an association, so that we aren't led down the garden path of, you know, thinking that something is causally related, spending lots of time and lots of money running these big RCTs to find out ultimately there is not an effect of using statins on dementia risk. So although it's null, I guess, yeah, it is a useful thing to know. A similar thing was done by some colleagues in Bristol and UCL, but this time using genetic instruments for antihypertensives. So you're kind of instrumenting the protein targets of blood pressure lowering drugs, essentially. And once again, we find very little evidence of an effect of antihypertensive use on Alzheimer's disease risk. One that is slightly more promising that I just really quickly wanted to highlight is a drug target study for metformin. So again, this is a study run by colleagues in Bristol, and they found essentially evidence that using 6 targets for metformin, there was an effect on Alzheimer's disease risk. So metformin reducing Alzheimer's disease risk in the general population, which is potentially interesting. And I think this might be my last slide. In relation to that last metformin MR study, which was a drug target MR, I just wanted, before I finish, to briefly touch on another causal inference method, which kind of supports those findings, again, like triangulation, another different type of study. And they used a different method, which is an emulated target trial. So this is one of the first studies which is just out to do this in the context of dementia. And I think this is a really exciting area of development, and I'm quite excited to see where this goes. But I have to confess, I know absolutely nothing about emulated target trials, so please don't ask me to explain in any great detail what they've done. But I think it's super interesting. So this one is for diabetes, and they are essentially creating almost like a pseudo RCT within a big cohort study or a big healthcare database. This one is the Kaiser Permanente database in the US. And they investigated the association of terminating metformin treatment for reasons that are not related to kidney dysfunction, which is quite common for metformin treatment, but can cause confounding. So people that terminated metformin treatment for reasons that weren't related to kidney dysfunction, and how that related to dementia incidence. So they essentially found that early terminators of metformin who were matched to routine users of metformin of the same age and gender, and who had had diabetes for the same amount of time, those early terminators showed increased dementia incidence. And they also found that it was largely independent of changes in their HbA1c level and insulin, which kind of potentially suggests some off-target effects of metformin. But yeah, I think super interesting that 2 completely different study designs have suggested that metformin could potentially be protective for dementia, and we should definitely look into that more. Okay, so I wanted to just give some kind of take-home conclusions, because I know I've just bombarded you with lots of stuff about lots of different risk factors. So on the whole, I would say education looks to be protective, but I think we are really just at the start of trying to figure out why it's protective and how we could kind of translate that into some kind of public health intervention to reduce dementia burden. For CVD risk factors, I think the evidence is super mixed. CVD risk factors have stressed me out for a long time, because it seems to just be a complete mishmash of evidence across different study designs, and getting any sort of answer for any one of them is super tricky. And I'm wondering if that is a reflection of this heterogeneity. So things like hypertension maybe being more important for vascular dementia and less so for Alzheimer's disease, but we're looking in these mixed samples. Evidence looks promising for metformin. And yeah, I guess the conclusion for me is just that dementia is really flipping hard to study, and we still don't know much at all. I think we have a really long way to go in understanding the causes, but we are making a good start, and yeah, people really seem to care about it. So I'm hoping that, yeah, the only way is up. That's the end of my waffle. I've got some references here because I think it's been recorded. So if anybody wants to go back and look at any of the studies that I've mentioned, those are the links. Thank you.
Thank you, Emma. That was an absolutely fantastic overview, and there's been loads and loads of questions in the chat and Q&A. So I think without further ado, I'll just begin to maybe navigate through some of the chat. We'll clear out the chat maybe and then go into the Q&A. So there's a question just going in reverse order on the chat questions. if the Mendelian randomization drug target trials on statin and hypertensive were done on vascular dementia rather than AD, would that potentially give us results?
I think that's a really interesting question. And I have my own theories, but they're absolutely not in any way evidence-based, because we don't have currently a big GWAS for vascular dementia. That is, as Park mentioned at the start, that's the kind of topic of my UKRI Future Leaders Fellowship. So we are in the process of genotyping 3,000 to 5,000 brain samples where we have neuropathological evidence that they have some form of vascular dementia. And the idea over the next 3 to 4 years is to essentially replicate what we have done in the Alzheimer's disease sphere, where we have these massive GWAS studies that we can do these drug target MRs or any MR in the vascular dementia space. Because my thinking is that I don't think it is going to be a surprise to anybody that hypertension is associated with risk of vascular dementia, because it's essentially, we know that is a causal pathway. So I absolutely think that it is plausible that we would start to see different results for some of the drug target MRs, particularly statins and antihypertensives. Not sure about metformin for vascular dementia, compared to AD, but we just don't know yet. Like I say, we've got so far to go, but we're on it. We'll get there.
And that links to the previous chat question. Does this all relate in terms of, for example, hypertensive medication pathways? Is that all somehow linked to inflammatory states artificially regulating the immune system, or are we simply not that far along in terms of the pathways for why these associations may or may not be seen?
I think before we can start unpicking those kind of mediating pathways, we firstly need to be identifying a causal effect. So I think to use things like multivariable MR, or even, like using basic science, doing experiments, we can only really start unpicking those mediating pathways if we have evidence of an effect to kind of unpick in the first place. But everything that we are seeing, at least in MR context and in most trials, is that for things like hypertension, there is no causal effect on dementia risk. There was that Sprint Mind trial, which I think, is providing evidence that it is potentially linked to dementia somehow. But my theory is essentially that we are looking at these really heterogeneous samples, and until we start looking at, Alzheimer's versus vascular, and we have these kind of cleaner samples to look at, we're not going to be able to unpick those specific mediating pathways. But I think once we have this big vascular dementia GWAS and we can compare the two, we can start, unpicking whether it's likely to be inflammatory or immune function pathways or something completely unrelated. Yeah.
And that relates to the sort of most recent question in the chat. How big does all of these samples for MR and GWASs have to be useful? I guess as big as possible is one answer, but I don't think we're quite there yet, really.
Yeah. So I guess for MR, MR is typically very underpowered compared with observational studies. I know Park doesn't like the term observational versus MR, because essentially MR is an observational study, right? We're not changing people's genotypes, we are observing them and looking at outcomes, but because they are random, I guess we try to distinguish between the two. But genetic variants for any given trait typically explain a very small proportion of the variance in a phenotype. So the PRS for Alzheimer's disease, for example, depending on the sample you look in, will explain between 7 and 10% of variation in Alzheimer's disease. Because it's explained in such a small amount, we need really big samples because our statistical power is quite low. So just to give some context, for the biggest Alzheimer's disease GWAS that we have, we're on to about 90,000-ish cases, and I think about 150,000 controls. But it's worth noting that for most of the Alzheimer's disease GWASs that we have, we've gone down this path of including what they call proxy cases. So rather than taking people that have had a clinical diagnosis of Alzheimer's disease, which ideally we want to do, we are taking people from the UK Biobank who have reported dementia status for one or both of their parents. So if the Index UK Biobank participant says, my mum or my dad had dementia, we will include that UK Biobank participant as a proxy dementia case. So there are different camps of people about that kind of think that that's a good idea versus it's a bad idea. But I think ideally we want to be boosting our sample sizes as big as possible with clinical dementia patients.
Thank you. I'm just sorry if you heard me swear, my internet connection just dropped. So next question, gene editing therapy for ApoE4. Could we be thinking about CRISPR-Cas9 to modify ApoE, which is quite a question.
Love that question. We've been thinking about this a lot. We're actually in the process of writing up a paper around intervening on ApoE, and it's interesting that you've gone to ApoE4 because some of our thinking around this is in relation also to ApoE3. So although ApoE3 is the most prevalent, we are still seeing an increased risk of dementia with ApoE3 carriers in relation to ApoE2. And we've worked out the population attributable fractions for if we could get rid of all ApoE3 and ApoE4. And essentially, if we could make everybody E2 carriers, we would prevent over 90% of all dementia cases in theory. So I think it's really interesting because we typically think of these things as not being modifiable, but we know now that they are. One key thing to think is that E4 is, so E2 has shown, although it's protective for dementia, shows detrimental effects for other things like respiratory disorders and I think also some liver diseases. It's the same with E4. So although it is detrimental for CVD and dementia, we are also seeing some protective effects of E4, which makes sense from a selection perspective as to why we still have, so many E4 carriers within the population. So I think it's a super interesting question, and in theory sounds plausible, but I think we would need to really understand the implications of wiping out a form of a gene sort of downstream.
Yes, indeed. So another question, to what extent does general cognitive function confound diagnoses, particularly for some cognitive tests such as the Montreal Cognitive Assessments, where you have to score below a certain threshold? And do other methods typically consider both loss or change in cognitive function and initial cognitive abilities?
I think there's quite a lot to unpick in that question. If I understand correctly, I guess you're saying, does somebody's kind of cognitive function affect their ability to perform on the test and affect whether they get a diagnosis? I'm not sure if I've understood that correctly, but we do see that in practice. So we definitely see that for the same level of pathology, people that have greater levels of educational attainment tend to do better on the cognitive tests and are less likely to be diagnosed, or they will be diagnosed much later. So I guess there is thinking around educational attainment providing compensatory mechanisms that enable people to function much better and do better on the cognitive tests. So even though they have dementia and they have the same level of pathology as somebody else, they are getting diagnosed much later. We do see that. And yeah, I guess it depends on the age that you're looking at your sample, because I think ultimately the later you go, the more likely it is that everybody would have had their dementia picked up. But yeah, I think that's definitely something that we have to think about.
So yeah, and there's a couple of questions related to that about whether the pathway by which education and or intelligence influences risk, and whether you're creating a bank balance of neurons that are very well exercised that then are healthier later in life. Is there any thinking or thoughts around that mechanism in the literature?
I think currently not. So there are two kind of thoughts. There's two avenues of thinking. One is around this kind of brain reserve, which is that physical thing that you just talked about with neurons. So is higher educational attainment or staying in school for longer doing something physically to our brains, creating more neurons, more denser networks? Is it increasing our brain volume so that when we do have atrophy, we have further to go before we reach that critical threshold of impairment where we would get a diagnosis? Versus cognitive reserve, which is less around the physical aspect and more around those kind of compensatory mechanisms that make you more able to function day to day, and therefore are less likely to get a diagnosis. there has been some work done. So Roxana, the PhD student that did the FEWAS, looked at the Alzheimer's polygenic risk score and an educational attainment polygenic risk score in relation to brain structures. And there wasn't a lot of evidence to suggest that education and intelligence were affecting, you know, brain structures physically, but it's really limited because we did this in the UK Biobank and it's just structural MRI. So we have volumes of, you know, cortical volumes. We aren't looking at, you know, neural density or, you know, neural pathways. There's no functional element to it. So I don't think it rules out the brain reserve hypothesis, but from what we've seen so far, we don't have evidence of that. I think we just don't know yet. I think we are at a point where we're pretty confident that educational attainment is causally protective because it increases cognitive function, and kind of innate intelligence / cognitive ability is protective. And the work really now starts in figuring out why. Like, does it matter if you're going to school, you know, in your childhood and adolescence versus if you go back to university in your 40s? Would that have the same protective effect? What is it about education? Is it like the being in that environment where you're being challenged and stimulated, or is it, you know, doing something physically to your brain? Like, I think we just don't know yet what it is about education that is protective, and I don't even know how we begin to go about figuring that out.
Thank you. And that links to a question in the Q&A about genes or other factors that might protect people with adverse ApoE4 profiles from developing dementia, if there's a connection with immune regulation as people age.
Yeah, so that is a really new area of research. We actually just have a grant proposal in around this at the moment, which is looking at epistasis. So are we seeing other genes that essentially modify the effect of ApoE in whatever form on phenotype? So the thing that we've been looking at is molecular phenotype, so proteins and metabolites, but we have also, like I said, seen that ApoE4 modifies kind of associations between observational phenotypes like smoking and physical activity with dementia risk. So I think the answer currently is we don't know, but it is an area of ongoing research. And I absolutely think, you know, we know that that exists for other diseases. I think it's highly unlikely that it wouldn't exist for dementia. Can we find other genes that essentially mitigate the detrimental effects of ApoE4? And if we can find those genes, are we able to mimic the effect of those genes in people that don't have the modifier by, you know, targeting a protein to have that same downstream effect? So epistasis is quite interesting for things like drug target, repurposing of drugs to essentially mitigate the effect of ApoE4 based on the effect of another gene. So yeah, I think it's absolutely plausible. I think it's really unlikely that that wouldn't exist. We just need to find it.
And just with 2 minutes left in the hour, just a couple of quick fire questions. What was the social intervention in the FINGERS trial? Was one of the questions.
So FINGERS is a multi-domain 2-year trial where they randomized people to a lifestyle intervention. So it was nutrition and exercise with cognitive training, and then intensive monitoring of their metabolic and vascular risk factors. So things like hypertension, cholesterol, etc. And then they did that for a period of time, compared their cognitive function at the end, and found that the intervention group did have some cognitive, they did better cognitively than the control group, but it is ongoing. So I think they've got another 7 years of follow-up planned where they'll start to see whether they have a difference in actual kind of hard endpoints. So are we seeing fewer dementia diagnoses in the intervention arm?
And I'm afraid we won't get to the question of whether dementia helps end the Cold War, but there is a question about whether risk factors like obesity, alcohol use, high blood pressure are maybe downstream of low physical activity, and maybe there's sort of different pathways that maybe come back to one or two small causes or singular causes, which I guess is what you're trying to unpick, and it's not easy to answer that.
No, and I think observationally we have done that and we see it, but then we're back to worrying about the issues with confounding and reverse causation. Yeah, I think trials are a really good way of unpicking those pathways. Like the FINGERS trial is a really good way of looking at that, but they're just really hard to do and they're really expensive. So yeah, in summary, everything is hard.
Everything is indeed hard. Emma, thank you so much. There's been a couple of comments about to congratulate you on an excellent, clear talk. Zero waffle, despite what you said. So for those of you joining, the talk will be uploaded very soon to YouTube. It'll also be accessible to the CPM website. So once again, just thank you, Emma. It was a fantastic talk, and we really appreciate your time in coming. So thank you.
Thank you.
Thank you very much. And thank you, everyone, for joining.